Hello Bio. We're going to continue on with viruses, the second part. And remember, we ended up with this picture last time, and now we're going to start off by looking at the two specific ways that viruses will infect different types of cells. So the first one we're going to look at is called a lytic infection. So in a lytic infection, a virus enters a cell, it makes copies of itself, and it causes the cell to burst. Now another reason why this is called the lytic infection is because another word for burst is lyse, L-Y-S-E, so that's where this gets its name from. So the steps of the lytic infection, so first they kind of use the bacteriophage as an example, but this can happen in other, and it could happen inside human cells as well. So the first is a bacteriophage will inject its DNA into a bacterium, and then the bacteriophage DNA forms a circle. So those are our first two steps, and that's actually the same in both lytic and lysogenic. So then the next stage is the bacteriophage takes over the bacterium's metabolism, meaning that it basically takes over how the cell is going to be making its energy, and it causes the synthesis of new bacteriophage proteins and nucleic acids. So instead of making new things to help out itself, it's making new virus parts is basically what it's doing. It's making parts to make up a virus. And then after a while, these different virus parts come together and they assemble into complete bacteriophage particles. So again, they go from just these floaty pieces to actual fully assembled bacteriophage. And then the bacteriophage enzyme will lyse the bacterium's cell wall. Again, that's where I said that lyse just means kind of cut open or burst, releasing the new bacteriophage particles that can attack other cells. So all these that just popped out are going to go do the same thing to other cells. Now in this picture we can only show like 1, 2, 3, 4, 5, 6, 7 viruses coming out of the cell, when in all reality it could be upwards to hundreds or even thousands of viruses coming out of a single cell going to infect more cells. So then our next type of infection is called lysogenic infection, and this is when a host cell will make copies of the virus indefinitely, meaning that the host cell will just constantly make new types of our new viruses. So in a lysogenic infection, a virus will integrate its DNA into the DNA of the host cell, and the viral genetic information replicates along with the host cell's DNA. So in our first one, it only made the virus's DNA. However, in this one, it's going to actually continue to make its own DNA as well as making the viral DNA. So how this actually starts is that it begins the same way as the lytic infection. So the bacteriophage will inject its DNA into the bacteria, and then the DNA will form into a circular pattern, and then the viral DNA becomes embedded in the host DNA, and it becomes what's known as a prophage. So this is a new word. Prophage is just a word that we use to describe that the fact that the virus's DNA has actually been incorporated into the host cell's DNA. And then the steps that kind of go in there is that, okay, and that's kind of the prophage. The bacteriophage DNA inserts itself into the bacterial chromosome, what we just said. And then the bacteriophage DNA may replicate with bacterium for many generations. So it's kind of, it can just kind of keep going along, keep making, or keep copying its DNA, and not really doing anything except just it'll keep making its own DNA. And then the bacteriophage DNA can exit the bacterial chromosome and then will enter the lytic cycle. So over the course of these generations, you can see that purple, the purple part is reproducing, and it's just constantly passing on the virus DNA to the next generation, until in fact it's time for it to enter the lytic cycle. And so that's how they reproduce. That's kind of how they infect our cells. Now we're going to look at a different type of virus known as a retrovirus. This isn't a virus that kind of goes back in time. It just does things a little bit backwards. So retroviruses contain RNA as their genetic information instead of DNA, and when they infect cells, they make a DNA copy of their RNA. So they go backwards. Usually we go DNA to RNA, they go RNA to DNA. And then this DNA is inserted into the DNA of the host cell. And an example of a retrovirus, one of the most famous ones, is the HIV virus. And it does the same thing. It makes its DNA out of RNA, which is backwards. So and that's what I just said. A retrovirus's genetic information is copied backward from RNA to DNA, and the viruses that causes AIDS is a retrovirus. So I got a little ahead of myself there. And so viruses in living cells, so this is kind of the debate on whether viruses are alive. So viruses must infect a living cell in order to grow and reproduce, and they take advantage of the host's respiration, nutrition, and all other function of living things. So they aren't necessarily alive themselves. They almost hijack the life of the living cells. But again, some people consider that to be alive. And again, we are going to make our own decision. And viruses have many of the characteristics of living things. So again, this is kind of looking at both sides of the spectrum here. After infecting living cells, they can reproduce, they can regulate gene expression, and they can even evolve. So these are things that we kind of associate with living things that viruses can do. And because viruses are dependent on living things, it seems likely that viruses developed after living cells. So what they're saying is it wouldn't make much sense that viruses have to have cells to survive if they didn't come after cells. So if they were before cells, what were they living on? It pretty much would be impossible for them to live unless they came around after cells. And the first viruses may have actually evolved from genetic material of living things, so they could have come along from the DNA or RNA of living things, and they have continued to evolve over billions of years, and they kind of do that every year, like with the flu vaccine. That has to change each year because each year the flu virus mutates and it changes and it evolves. So that's it for viruses. Let me know if you have any questions.